Welcome to another question series. Today we'll be looking at lipids and proteins, looking at the structures, classification functions, and tests for lipids and proteins. So we'll be looking at several questions involving this class of food. So the first one, we'll be looking at lipids are primarily characterized by being soluble in organic solvents. So that's how we classify lipids. They are not soluble in water, they are insoluble in water, but they are soluble in organic solvents. So the answer to that question is C. Fatty acids are long chain carboxylic acid. Long chain carboxylic acid. There's a video on my YouTube channel that's focused on fatty acids and lipids. So we have C. We have C in carboxylic acid, then we have long chain carboxylic acid. Usually you can see start from 4, 6, 8, 10, 12, 30. So they are long chain carboxylic acid. So this is whenever I say molecular, I know that it's actually a fatty acid. Now saturated fatty acid contains no double bond. No double bond, only single bonds. No double bonds. Answer to that question is C. There is no double bond at any point on the fatty acid chain. Unsaturation contains one or more double bonds. You cannot say it contains only single bonds. No. You cannot say no. The answer is one or more double bond. If it is one double bond, carbon-carbon double bond, we'll say it is mono-unsaturated. Mono-unsaturated. If it is more than one, we'll say it is poly-unsaturated. Poly-unsaturated. So it's monounsaturated and polyunsaturated. So you see something like MUFAS, MUFAS, monounsaturated fatty acid, or PUFAS. You see PUFAS, polyunsaturated fatty acid, when they have more than one double bond. Oleic acid is an unsaturated fatty acid. Now, is it a mono or is it a poly? It's a mono-unsaturated fatty acid. It has only one double bond at position 9. Only one double bond at position 9. So if you have the fatty acid like this, we have 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So we have 18 carbon atoms. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. 18. So we'll now look at the 9th position, starting from the carboxylic carbon. 1, 2, 3, 4, 5, 6, 7, 8, 9. So the bond will be between 9 and 10. This is carbon number 9 and this is carbon number 10. So the bond between carbon number 9 and 10. So you can say this bond now is trans, but the oleic acid is actually a cis kind of bond. This bond now is called a trans double bond. Why? If we have the same fatty acid, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. So here we now have 1, 2, 3, 4, 5, 6, 7, 8, 9. This is 9, this is 10. So the double bond is here. 11, 12, 13, 14, 15, 16, 17, 18. So this kind of bond is called a cis bond. It's called a cis bond, and that cis bond introduces what is called a kink. It introduces what is called a kink, like a K-leg. So it is a monounsaturated fatty acid having the double bond between 9 and 10 carbon atom. So we'll give it the lowest possible number, which is number 9. So usually they will tell us this is cis delta 9. That is delta 9 tells you oleic acid, tells you that the position is at carbon 9 and the orientation of the double bond is a cis double bond. So that is a monounsaturated fatty acid, oleic acid. Waxes are esters of fatty acids and long chain alcohols. Long chain alcohols. So when we say esters, it means that esters are formed from carboxylic acid and alcohols. So when carboxylic acid reacts with alcohols, esters are formed. So you see in nature like this. This is the fatty acid. This is this one will have long chain. This one will also have long chain. So this is the alcohol. This is the fatty acid. So together they form esters. So these are waxes. Now, function of waxes is waterproofing. That's what plants use to, prevent water loss. Is to prevent water loss. So we have sterols possesses 4 fuse rings. 4 fuse rings, also known as the cyclopentanoperhydrophenanthrin. Now this is how sterol looks like. The first ring, the second ring, the third ring, and the fourth ring. So this is the core structure of sterols. So any molecule that you see that have this core structure, you know that they are talking about sterols. And that is where we have our steroid hormones from. The androgen, the estrogens, the progesterone, the estradiol, the estrone, even the testosterone, the male hormone, is synthesized from this core structure. So it's actually 4 fuse rings. 1, 2, 3, 4. 4 fuse rings. This is called the steroid nucleus. Cholesterol is a sterol. It does those 4 fuse rings. It's a member of the sterol class. Terpenes are formed from repeating unit of isoprene. Isoprene. So terpenes is also a class of lipids that are produced from isoprene units. So this is our isoprene. Isoprene is 2-methyl-buta-1,3-diene. Buta-1,3-diene. So buta means 4 carbon atoms. 4 carbon atoms. So 2-methyl. So there's a methyl group here, CH3. So 1,3-diene means there is a double bond at between 1 and 2. There's a double bond between 3 and 4. So this is 2-methyl-buta-1,3-diene. Now this carbon will have how many hydrogen? 2 hydrogen. This carbon will have 2 hydrogen also. This carbon will have 1 hydrogen. So this is isoprene, which is 2-methyl-3-diene. Di means 2 double bonds. Diene is the building block for terpenes. Phospholipid contains fatty acids and phosphates. But now, the actual sense of phospholipids is not just fatty acid and phosphate. It is fatty acid esterified to a glycerol and also having a phosphate that is also esterified to the glycerol backbone. So we have phospholipid in this nature. 1, 2, 3 as a glycerol. Now this glycerol is an alcohol, so it will be esterified to a fatty acid. Fatty acid 1, fatty acid 2, then this one so this is a phospholipid. This is a phosphate group, the fatty acid, all esterified to this backbone called glycerol. Phospholipids are amphipathic in nature. They are amphipathic. This amphipathic means they are both water-loving and water-hating. So it is the phosphate group that gives it the water-loving properties. And there are about so many types of these phospholipids. We have the phosphatidylinositol. If an inositol is attached to the phosphate group, we have phosphatidylserine. If a serine is attached to the phosphate group, inositol. We have the phosphatidylcholine. If a choline is attached to the phosphate group, we have phosphatidylethanolamine. If ethanolamine is attached to the phosphate group. So the phosphate group gives it the water-loving properties, while the glycerol backbone and the 2 fatty acid gives it the water-heating properties. So they are amphipathic. They can interact with water and can also interact with non-aqueous or organic solvents. So they have both polar head and non-polar tail. So the phosphate group gives it the polar head. Okay, so the major role of phospholipid is membranes formation, which is the formation of phospholipid bilayer. They form phospholipid bilayer that enables us to have a biological membrane. So do not forget to click on the subscription button and also on the notification bell for more information to get updated whenever we post a new question and answer series.